from a feedlot. Gram-negative rod was isolated from runoff from a dairy farm. Ooh, gram-negative rod. All right, so all of those things tell you gram stain. So on your dichotomous key, remember your first choice, the first decision you've got to make is it gram positive or gram negative, and is it a rod or a cocci? So we have covered those. Anyone have any questions of anything that they have read on any of the practice questions that wasn't covered on gram stain or shape? All right, so the next decision you have to make on this, on the gram-negative side, and remember, most of your unknown word questions will be from the gram-negative because that's where most of the genera are. Most of the genera are gram-negative. So the next decision that you need to make is...
Before you erase that, can I ask you a question?
Yeah.
On number 4, after it says lipids outside the cell wall, does it calcium?
Calcium ions.
I thought that calcium...
Endospore is calcium ions.
Okay, endospore. Okay, because I always took it as... So, I always took it as the calcium meant it was endospore positive.
It does.
Okay.
It also means it's gram-positive. Because all endospore positives are gram-positive, just like all acid-fast positive are gram-positive. Good question. All right. What? Understand?
Oh, I'm gonna go get the question.
Oh, yeah. Good idea. The next thing we're going to talk about is how to find your glucose values. And that's kind of easy because there's not many answers. So, to discover your glucose, sorry I'm going to colors, but I'm running out of marker. Who would have thought? I didn't think about getting a marker. All right, so where can you find your glucose? Of course, the number one place to find glucose is phenyl red glucose. Broth. So I can disguise that as calling it the pH broth. I can also disguise it by calling it the first of the 3 major broths. What are the 3 major broths in order? Glucose, lactose, and mannitol. So if I say the 3 major carbohydrate broths, and I say it was +2 - 1, that means these 2 were positive and that one's negative. If I say +3, they're all. If I say -3, they're all negative. How else can I disguise a glucose result? I often use PR dextrose. Dextrose is the old name and the traditional name for glucose. So those are the ways I can disguise that in a word question. Where else could you get your glucose result? And that is Kligler's iron. Kligler's iron slant deep. And if you recall, on the Kligler's iron slant deep, you have a test tube, and it has auger in it. Oops, let's make it a little bit more pronounced. Like that. And in the slant, the value is lactose, and in the butt, the value is glucose. And the pH indicator is phenyl red, so yellow equal positive or acid equal positive. And red equal negative or neutral, let's say alkaline. So the glucose result can be found in the butt of the tube, and it has to be yellow to be positive. If it's red, it's negative. And remember, I can also tell you that whether or not it has gas. Remember, it can have gas by pushing the yellow up and leaving a space here, or by cracks in the agar, or by bubbles. Now, it can't have gas unless it's positive. So if I disguise the glucose result by saying the Kligger's iron was red and yellow, no, the Kligger's iron was all three colors, then you'll know that one had to be red, one had to be yellow, and the other one had to be the hydrogen sulfide was black, because that's the three colors of it. So now, which one was yellow and which one was red? Only way to find out is know your lactose test. If your OMPG was negative, that meant this is red and that is yellow. So another test will tell you. So remember, here you have, I say, the Kligger's iron was three colors. You know those colors. Black means it's positive for hydrogen sulfide. You know that red means that one of the sugars was negative, and that the other one was positive. Now, how do you know which is which? You look for another test. If your lactose was positive, then your glucose is negative. If your lactose is negative, then your glucose is positive. What other tests are you? There are this many lactose tests.
Are they always opposite?
No, you got to get three colors. So if lactose is positive, or that means the yellow is the lactose, which means that the other one is the negative. You got to have three colors. I said it was three colors, so one of them's yellow and one of them's red. So lactose will tell you what the other one is. So what are your lactose tests? Number one, the best one, OMPG. If it was yellow, if it was white, negative. What's the next best one? ER lactose, in red lactose broth. Same thing, except that the negative is red or orange.
How do you disguise OMPG?
Just by saying the test done from a dioxic slant. Name some other lactose tests. EMB, Tectoin, McConkey's,
Litmus?
what?
Litmus?
Yeah, litmus milk. Anybody think of any others?
Should be 8. One.
What?
I said there should be 8, correct? 6.
What have we got?
EMB, MR? No.
Oh well, those are the ones I use. Remember, this would be pink if it's positive. What would this one be? Hot pink on orange, or
Hot pink on hot pink.
hot pink on hot pink. Hot pink on orange, or hot pink on hot pink. Hot pink on hot pink is heavy. This is heavy lactose, positive. This is a moderate lactose, positive. Salmon is positive for lactose on hectoin, and purple on purple is moderate. Positive on lactose on EMB, and green on purple is heavy. So these are all the ways to find out a lactose result. And this is all the ways to find out a glucose result. And remember that this is taken from the KIA, or the test that exhibits dioxic growth or catabolic repression, which is also called catabolic. What does that mean? Bacteria always eats glucose first. When there is no more glucose, then it will digest lactose second, which means you will have a growth curve and followed by another growth curve. This one for glucose and this one for lactose. That's what's meant by dioxic growth, and remember the only test we have that has both glucose and lactose in it is KIA, Gleager's iron. Okay, any questions about how to find glucose or lactose? All right, let's talk about something easy. How to find hydrogen sulfide. Hydrogen sulfide, only two tests will tell you hydrogen sulfide, although there is a couple of tricks. Actually, three tests, sorry. If you look on the gram negatives, about the third or fourth question is hydrogen sulfide. Otherwise known as H2S. So what are the tests? The number one test is KIA, black anywhere. So if I say there was a black dot, that's enough. Doesn't have to have a black tube. But if some of you did it, you'll notice that if you do have a lot of black, sometimes it covers up all the glucose. So you could have a KIA that looked like this. This would be red, and everything down here would be black. So you wouldn't have your glucose hidden by the hydrogen sulfide. So that means another test is going to tell you your glucose result. All right, so what else can give you a hydrogen sulfide result? And that's peptone, iron, deep. It can be brown or black. It's a deep. We usually have it, but this year she didn't make it. It's just a deep that you stab. And if along this grows a line of growth that appears brown, in reality, if you dissected this out, you would see it's black, but because it's coming through a yellow auger, it looks kind of brownish. That's hydrogen sulfide positive. What's the only other way to get a hydrogen sulfide result? Visibly, hectoin. The hectoin auger plate has black crystals. In the growth line. In other words, where you put the line on the plate, there is a piling up of the growth, and there are little dots of black in there. that will tell you it's hydrogen sulfide positive. Alright, what's the final way I can hide a hydrogen sulfide results? I'll say a gas detected by a precipitate. Because when hydrogen sulfide is in the presence of iron, it's a precipitate, which is black. So that's the actual chemistry of what's going on. The gas reacts with the iron in any auger and makes a black precipitate, and that's where you get the black color. But if I didn't want to say black, I would just say a gas detected by a precipitate. All right. So that's all the hydrogen sulfide. Any question about hydrogen sulfide? The next thing we're going to cover is citrate. There's only 2 citrate tests.
Mr. Hicks,
Yeah.
before you move on the red color, you said salmon color is positive, right?
For lactose. That's
Yeah.
it.
But in our cheat sheet, it says blue-green is salmonella, green is shigella.
That's
the only-
growth. The colonies on the green auger. Might as well get rid of it right now. What does hectoin tell you? 1, salmon around the growth. Equal positive for lactose. Crystals, black crystals or any crystals, in the growth equal hydrogen sulfide positive. Colonies that grow blue-green. What's another word for blue-green in English?
Teal.
Teal. What's another one? Oh, turquoise. On green auger. What is it? You just read it. What is it?
Salmonella.
Salmonella. What if it is colonies that grow green on green auger? What is it?
Shigella.
Shigella. So hectoin can tell you a lot of stuff. And what's it? How do I hide hectoin? I call it the green auger plate. Which takes us to the other green auger. Green auger slant D is Simmons citrate. And remember that any blue, a dot of blue, a whole blue tube, half a blue tube, any blue equal positive for citrate. What's the other citrate test? Kosers. It is not as good. If they conflict, go with Simmons. Kosers, citrate, raw. The only thing that's in there is saline and citrate. If your bacteria can eat citrate, they will divide and make little clumps. Those little clumps will settle to the bottom of the tube. So when you pull it out of the incubator, you have to thump it. But when you thump it, you add gas or air to it, and you can see bubbles. So you have to distinguish between the bubbles coming up after you thump it and a precipitate or a little flakes of something. So cloudiness or particulate. Equal positive for citrate on this, and clear equal negative for citrate. What's negative on Simmons? Green. So that takes care of all the citrate tests. Anyone have any question about citrate? Alright, so on our gram negatives, we have covered gram stain, we've covered shape, we've covered glucose, we've covered citrate, we've covered hydrogen sulfide. The next thing that we want to cover is oxygen usage. So we're going to talk about thioglycolate broth and candle jars and incubators and a slant with mineral oil. So the next thing that we're going to discuss on the word problem hints, set number 5 and 6, right? Okay, so this will make this one 7. Oxygen utilization. And that is usually the thioglycolate broth, which is fragile. I might say, the broth that cannot be shaken, the broth that should be handled very carefully, the broth that has a 1-inch pink layer at the top. Will all be things to hide thioglycolate broth, to keep me from actually saying the word thioglycolate. Or I'll say the test that tells you whether or not your bacteria has a modified Krebs cycle. So all those things are to hide the oxygen utilization test. And remember, there are 2 aerobes. The obligate, meaning must have oxygen, aerobe, and the microaerophilic, which must have oxygen, but cannot have it at 20%, which is what we would have in this room. So these microbes, here's the fluid, these microbes what's it, 8:05? I don't answer phones. They don't leave a message, they didn't need to talk to me. Alright, so if there is cloudiness at the top, and it is crystal clear, some of you people don't know what cloudy looks like, below, it's an obligate aerobe. If it has a narrow band of cloudiness, but clear above and below, it's microaerophilic, and these are the soil bacteria. I might disguise it by saying this microbe was isolated in the first 3 inches of the soil. So you would know its utilization is aerobic microaerophilic. All right, the next ones are the bacteria that can take it or leave it, and they're the facultatives. And there are 2, but one of them is overwhelmingly the majority. And that is cloudiness at the bottom with a wisp tampon string to the top. And this is facultative anaerobe. Remember, facultatives can use oxygen. They have a modified Krebs cycle. and they can choose or choose not to, and most of them live in your intestine like E. coli and don't use oxygen usually, but they can, so they prefer not to have. So this is facultative anaerobe. And there are very few microbes that have heavy at the top with a little string all the way down, and that's facultative aerobe.
What were you saying was gonna be pink?
When the broth, when you get it, will look like this. It will be a brick-colored broth, and at the top there will be a layer of pink that is about 1 inch. This is where oxygen has penetrated the liquid from the preparation process, and that's where your aerobes will grow. Now remember, if you slosh it around too much and the pink gets down to this much, the tube cannot be used. All right, so those are the facultatives, and then we get to the anaerobes. These do not have a Krebs cycle in their plasma membrane. And there are 2 of them. They only use glycolysis, fermentation, and a modified electron transport or a minor electron transport system. And they are equally cloudy throughout because oxygen doesn't matter. So that's called aerotolerant, and we don't have any in ours, but I might give it on a word question. And you would know that that is a anaerobe. Or cloudy only at the bottom. Clear here, cloudy at the bottom, and that's obligate, must not have oxygen. Oxygen will kill this one. And these will live in a candle jar, although this one will live best. So how do you know it's an obligate anaerobe instead of one of those others that will live in a candle jar? Catalase test is negative. What else will grow anaerobes? A carbon dioxide incubator, or mineral oil on a slant. To grow an anaerobe, we take a slant and we put the bacterial on it, and then we cover it with mineral oil so it now has no oxygen, and it will grow. So all those are ways to tell the, if it grows on a tabletop, what is it? Aerobic or facultative, or aerotolerant?
CO2 incubator.
I can't hear you.
CO2 incubator.
Carbon dioxide incubator?
Yeah. What is that for?
Anaerobes? Anything that's killed by oxygen has to have a carbon dioxide incubator. Any cell in your body grows in a carbon dioxide incubator.
That's anaerobe.
Hmm? Remember, the cells in your body don't like oxygen, only your respiratory system likes it. Okay, so that takes care of oxygen utilization, which is used in both gram-positives and gram-negatives. Does everybody know how to hide an acid-fast stain result?
No.
Differential stain that uses a hot pink primary dye or a cerise primary dye or detects lipids is the acid-fast stain. What else hides an acid-fast stain? I can't think of anything else. All right, what hides an endospore stain? How would I tell people that the endospore stain was positive or negative?
Calcium ion.
Right, if we detected calcium ions in the solution, that would tell you it probably had endospores. It has to have rods, remember though, to be endospore positive, it has to have rods. Rods with holes or green ovals were detected. All of those things tell you endospore. So let's just put this up here for people that are writing it down. 8, acid-fast stain, positive. The differential stain, whose primary was hot pink, fuchsia, cerise, all of which is hot pink, or purple red, tells you that it is positive for the acid-fast stain, but remember, it's got to be a rod. So if I say that it had coxy and a few of them stained cerise
Negative.
and blue, it would not be acid-fast positive, because coxy can't be acid-fast positive, that's just bad staining. What if I said that there were dots of green in it? It doesn't mean it's endospore positive, that's dye debris. Dots are dye debris. Any dots mentioned would be dye debris. It would have to be a cell shape that was that color. What else can hide? Lipid, the stain that detects lipids. On the acid-fast stain, I'm trying to think if there's any other way I hit it. Stain, this microbe was detected from a patient's sample taken from the respiratory system. In other words, they were coughing up tuberculi phlegm. And yes, phlegm is a word. So all those are ways to hide the acid-fast stain. Now let's talk about ways to hide the endospore. To mislead you, be positive, detect calcium ions.
How does the phlegm let you know that it's acid fast?
Just a second. Okay, what was your question?
How does the phlegm tell you that it's acid fast?
Because the only microbe that's ever isolated from phlegm besides bloody phlegm is tuberculosis,
Okay, so
mycobacterium.
assume that it's tuberculosis, not like flu or something.
Yeah, I mean, if it's a color, it has to be a colored phlegm, but phlegm is assumed to be colored. If you're having a